Okay, so I'm going to shift gears just a little bit and talk about all of you know the brain is simply an extension of the retina, so what I'd like to do is focus back to the home port here and talk a little about up the eyes. Four things I'd like to walk out here after my talk thinking is that, number one, the leading cause of vision loss in industrialized nations is neovascular eye disease, which affects blood vessels and concomitant neuro and glial degeneration in the eye. Macular degeneration affects over 30 to 40 million people worldwide, and about 8 to 10 to 15% of those patients actually have vision loss from the disease. Diabetic retinopathy, again, we're thinking more as a neuronal degenerative disease, but has a very significant vascular component to it as well. And finally, the neurodegenerative diseases like retinitis pigmentosa, which are thought of typically as neurodegenerative, actually have a very significant vascular component, which is not simply atrophy of blood vessels because neurons are gone, but rather this crosstalk that you just heard about from the other speakers, failure in that crosstalk. We have ways to treat neovascular eye diseases now, largely VEGF antagonists. Unfortunately, only about a third of the patients benefit from these treatments, which means about two-thirds of them either stay the same or continue to get worse. So we still have lots of room to do something therapeutically. And at the end of the day, you don't really want to bash these blood vessels. What you really want to do is improve them and make them better. You want to improve the vasculature as well as the neurons. And this is why we began to think about 10 or 12 years ago now about using progenitor stem cells to approach this. I think stem cells or progenitor cells may be a way to do this, but how they do this is still open for discussion. As you heard earlier, there are a number of clinical trials in the eye using a variety of iPS-derived or vascular progenitor cells to treat these diseases. I think what's become clear is that most of the effects we're observing, at least to date, are not necessarily through cell replacement therapy, but rather through our paracrine effect, which you just heard Sean alluded to in his talk. Why do the eye get so many neovascular eye diseases? Because the eye is basically a bag of blood vessels. What you can see here is you've just stripped away the outer segment here of the eye, something called the sclera, and you see these vortex veins. If you go into the eye itself, you actually have a highly ordered structure. The retinal portion of this is over here. It sits on a layer of cells called RPE cells, which sits on a very rich vascular plexus called the chora capillaris. And the inside of the retina is lined by retinal blood vessels themselves. And if you do a cross-section through this, what you can see is that there's 3 layers of retinal blood vessels which permeate the entire inner retina. The outer retina, which are largely photoreceptors and RP, are subserved by this underlying chora capillaris. And again, the details aren't as important. I'd like you to keep in mind as we speak a little, as I continue with my talk, that there are lots of blood vessels here. Okay? And again, as I mentioned, historically, the way we've treated neovascular eye disease is by matching up supply and demand. That is, we lay down laser in the peripheral retina to destroy the peripheral retina in the hopes of preserving the central retina. If you decrease the demand by killing off half the retina, you can preserve what's left over here. These are abnormal new blood vessels. But laser is a destructive treatment modality, and I think the New Yorker magazine captured this very nicely. This cartoon, this gentleman just lasered his colleague's head, and it says, "For God's sake, Edwards, put the laser pointer away." So my point here is that this is a destructive treatment modality, and even though we do this in the clinic still, we prefer not to be able to have to do that. What we really want to do is to find better ways to stabilize the vasculature and perhaps normalize these abnormal blood vessels and restore normality. There are 4 basic ways where we've approached thinking about using progenitor stem cells for treating eye diseases. One is vascular trophic agents. That is, these progenitor cells themselves carry a variety of paracrine functions and molecules which we think help to stabilize and improve dysfunctional vessels. It turns out when you stabilize the vasculature, we also see a very dramatic improvement in retinal neurons as well, and I'll discuss that a little more later on. We can also, once we figure out how these cells target and where they go, one can think about loading them up with other types of trophic or static molecules, which may be useful. I'm not going to talk about that today, but we've done some work in that area also. And finally, the thing we all hear about mostly on TV and radios and other places is to use them as replacement parts. That is, can we replace defective neurons, glia, blood vessels in the eye by providing them with stem cell-derived whatever. Let me talk first about, I'm going to take you 2 stories from the lab. One focuses on the use of iPS-derived retinal pigment epithelial cells. And these RPE cells we know play a very significant role in what we call atrophic macular degeneration. And this is just what we call an optical coherence tomography, or an OCT. The nice thing about the eye is, unlike the brain, it's difficult to image directly. The eye we can look at directly through a variety of very neat tools. This OCT allows us to image the surface of this patient's retina and actually do microscopical sectioning, serial sectioning through the retina itself. And you can see there are splotches here where the retina has degenerated and areas we call geographic atrophy where the pigmented epithelial cells have disappeared. Fortunately, it turns out that you can take embryonic stem cells and iPS cells also, and one of the default differentiation pathways is into RPE cells. So it's pretty easy to make RPE. And Ray Lund and his colleagues at Albanese and others had described this many years ago. We are working with Yang Zhu here at UCSD and Shen Ding, who used to be at Scripps, who's now at UCSF. We're able to devise a method for deriving iPS cells, which eliminated 3 of the 4 Yamanaka factors, which have potential oncogenesis. And you heard about that earlier today, some of the downside of that. So we've used small molecules and OCT4 to generate our iPS cells. These cells can then be plated out once you get the iPS and stimulated to differentiate into RPE and other cell types in the back of the eye. We've succeeded in making macular glial cells and photoreceptors. We'll talk about that a little bit later. But for now, let me just focus on the RPE cells that we've made. The RPE cells themselves are actually really good RPE cells. They form this hexagonal array when plated out in tissue culture. Do we have another pointer? I think this is not working. I'm not sure who. Well, it works intermittently. They polarize by producing membrane proteins which are polarized to the basolateral apical surface, in this case, cis-retinaldehyde binding protein. If you feed them outer segments of photoreceptors, which is something they ordinarily do, they will phagocytose those outer segments. And interestingly, they also pump water. They'll form these little these little domes in tissue culture, which shows they're pumping water the way they're supposed to because of the water pumps that are present on the basolateral surface. Interestingly, or most importantly, when you take these iPS-derived RPE cells and you inject them into a rat model of RPE degeneration, something called the RCS rat, which resembles the kind of atrophy observed in patients with macular degeneration, what you see is in the uninjected eye, you get significant atrophy of the outer retina, or the photoreceptors are pretty much all gone. Vasculature is also degenerated quite significantly. The contralateral eye in the same animal, which has been treated with these, what we call the one-factor iPS-derived RPE cells, there's a good maintenance of the outer nuclear layer, and these photoreceptors actually are functional. Thank you. Okay, great, thanks. And you can see if you do electrophysiology on these, the cells that are injected with the iPS, we can actually do focal ERGs where you shine a stimulus on the area of the retina which actually has the iPS-derived RPE. You get a good response electrophysiologically. The contralateral uninjected eye or the spots in that same eye actually have very diminished amplitudes. So we've been able to observe that the iPS can be reprogrammed from keratinocytes and also blood cells now, of course, using only OCT4, OCT4 and small molecules. These cells are very good RPE cells. They look very much like healthy, young, fetal RPE cells. They are anatomically and functionally mediate functional rescue of animal models in which you have degeneration. And we're using this technology to deliver various trophic and static molecules to the other site of degeneration. I use this because these cells are sort of ear today, gone tomorrow, part of the analogy here. So this mouse has an ear, but it can be gone tomorrow. And the cells we observe, even in the human clinical trials, while some of these cells actually do take up house and set up shop in the back of the eye, many of the cells are transient, and there are not enough cells left to account for the functional rescue or anatomic rescue that we observe in the animal models and what limited information is available in humans. So the challenge is really trying to understand what these cells are doing. We have to understand better the targeting rescue mechanisms, and to do this, we and other laboratories have used a variety of approaches, including transcriptomics, proteomics, and more recently metabolomics, trying to understand what the rescue effect is. So we asked the question, if you inject iPS RPE cells into this RCS rat, and then analyze the rescued versus non-rescued rat, what are the differences? And we chose to use an untargeted global metabolomics approach, which was spearheaded by several people in the laboratory. And what we observed was the most significantly dysregulated class of molecules in the degenerate retinas were the fatty acid amides, okay? And one specific fatty acid amide, in fact, aurucamide, was the most significantly dysregulated. If you looked at the RCS rat, level of these molecules was significantly diminished when compared to wild-type rats. We then looked at iPS-derived RPE rescued RCS rats. And what we found was this much more closely resembled the wild type. We almost completely restored the level of fatty acid amides and aurucamide. And so we asked the question, well, what do we need the stem cells for? Maybe we can just put fatty acid amides and we can put aurucamide. And indeed, if you inject purified aurucamide into these animals, again, this is the uninjected eye. This is the RCS rat eye treated with aurucamide. And we see a very nice restoration of the outer nuclear layer. This just quantifies it. And if we do functional electrophysiology, we also see a functional rescue. So we were very excited. We thought we'd identify at least one potential pathway, and we chose to look at another area of interest in the laboratory, and that is the vascular rescue. That is, can we study the potential effects of endothelial progenitor cells, and by rescuing or stabilizing vasculature, could we similarly achieve a neurotrophic rescue effect, and what's the basis for doing that? And so let me see if I start seeing a flesh. We'll probably make it through, but if we don't, let me give you the punchline. Basically, what we're able to do is to identify and isolate and purify from adult bone marrow, and this is human bone marrow as well as animal bone marrow, human cord blood, populations of cells, which we call endothelial progenitor cells, which turn out to be largely myeloid progenitor cells. And these cells do one of two things. If you isolate the cells, in this case, these are from GFP transgenic mice, so all the cells that are injected are bright green, and the recipient retinas you're looking at here are green astrocytes. These cells will target to underlying glial cells, okay? And they'll either participate in formation of new blood vessels or form mosaic blood vessels, green being the injected progenitor cells, red being the endogenous endothelial cells, or they will take these perivascular positions. And perivascular vessels, which is the majority of vessels that we inject, are differentiate into microglia, and these cells differentiate into endothelial cells, both of which give a very profound rescue effect when put into models of ischemic retinopathy, which I'll describe for you in a few moments, or neurodegeneration. So let me spend the next couple of minutes talking a little bit about the specifics. We can select what we call lineage-negative cells from bone marrow. So these are not the cells that form the formed element of the blood, but rather what's left over. And the cells that are left over, we call lineage-negative, lineage with respect to forming red blood cells or lymphocytes. And these are myeloid cells, and they're largely myeloid progenitor cells, which display many of the markers which we associate with endothelial progenitor cells. If you compare the two populations of cells, they also express high levels of CD44, which is the hyaluronic acid receptor. I can't tell you what relationship hyaluronic acid has to this, but we can come back and talk about that later if you'd like. So again, the lineage-negative cells are enriched for EPC markers and also very high levels of CD44. If you take these cells, again, from GFP transgenic mice and inject them into retinas, these cells sit out in the back of the eye. If you come back and look later on, you notice that many of them are beginning to line up along forming new blood vessels. Mice are born without a retinal vasculature and form that vasculature during the first 3 weeks postnatally. They form a superficial layer at 1 week, a deep layer at 2 weeks, and an intermediate layer at 3 weeks, which is virtually identical to what happens to people during the last trimester of pregnancy. So it's a very nice model if you're interested in studying retinal vascular formation in association with neurodifferentiation in the retina. So what we observed was that basically you can inject these cells, again from GFP-enhanced mice, into a mouse in which the astrocytes are expressing GFP also, but regular GFP. And what you see is the injected cells all settle down onto this pre-existing astrocytic template. And as the vasculature forms, they will aggregate to form vascular networks in these mice, and these are patent blood vessels as evidenced by injection of rhodamine dextran. So very cool. You can inject these cells and they'll target to the astrocytic template, which is identical to what's utilized by endogenous endothelial cells, and participate in formation of blood vessels. What was really interesting and just really freaking remarkable is that if you inject these cells and then come early on, say postnatal day 1, before the vasculature, the outer vasculature is actually formed, and come back several days later, you find them on the surface of the retina. Again, this is the inside of the eye down here, this is the outside of the eye. You find the injected cells line up on that forming primary vascular plexus. But what's remarkable is cells have begun to migrate into the retina and take up positions identical to where the subsequent vascular layers will form, that deep and intermediate vascular plexus. So these cells know where to go and they know how to do what they're supposed to do. So what? So what this means is we can take a neonatal eye and perhaps incorporate some sort of stem or progenitor cells into this. But can you do this in disease conditions? So the question we asked was, if we stimulated the focal gliosis in an adult eye, that is focal proliferation of astrocytes or microglial cells, which is similar to what we observe in many of these neovascular eye disease states, would we get a similar targeting of these cells? And indeed, if you stain for GFAP, this is an eye that's stimulated with a laser to create a little scarring on the surface, or a needle, I'll just scratch it with a little needle. In both cases, you get local focal proliferation of GFAP-positive glial cells. If you inject those endothelial progenitor cells, they localize to the same area where the focal gliosis is, and they don't go to other areas. And they actually will eventually differentiate into and form blood vessels. So these cells target activated astrocytes. They'll participate in new blood vessel formation. It's dependent upon a number of molecules, which I don't have time to discuss today. And the real question, though, is will they provide trophic rescue under hypoxic stress? And the answer is yes, they will. So what we've done here is it's a model called oxygen-induced retinopathy. In this model, the mice, I won't go into the details, but the readout is areas of vasoobliteration, highlighted in yellow here, where blood vessels are no longer present, and massive neovascularization between perfused and non-perfused retina. And what happens if you treat these with these lineage negative CD44 high bone marrow cord blood derived progenitor cells, you see a virtual elimination of the obliteration and a highly significant reduction in the area of neovascularization. This is just quantified here to show you it works really, really well. So what are these cells doing? As I mentioned, most of these cells are not becoming blood vessels, but rather assuming perivascular positions. They go in expressing markers of myeloid cells and myeloid progenitors, and once inside the retina, they begin to identify markers which more look like microglia. So it turns out these cells differentiate these myeloid progenitors from adult bone marrow or cord blood, differentiate to microglia, and exert a very profound rescue effect. So in keeping with the theme of the other two speakers, we asked the question, well, if older bone marrow does this, does blood from younger individuals do a better job? So to do this, we isolated blood from cord blood cells, and also from cord blood cells. And these cells, turns out, differentiate into different macrophages, and these cells have the same kind of rescue effect in those hypoxic models as well as normal development. We've more recently looked at even more primitive cells, these what we call endothelial colony-forming cells. And again, these cells can be isolated and expanded into colonies, and again, injected into these models and show the same kind of rescue. We can do this in Zenofree media also, which has been done in collaboration with a group up in Vancouver. So we now have ways to isolate these cells from cord blood, which will generate these progenitor cells in Zenofree media with the potential for applying these in the clinics. As I mentioned earlier, in models of retinal degeneration, we have vascular degeneration. You get very profound rescue of vessels with this, but you also get very profound neurotrophic rescue. This is a model, something called RD10, mice in which the photoreceptors degenerate along with blood vessels, and when treated with these progenitor cells, you get the same kind of neurotrophic rescue. And these effects can also be enhanced by overexpressing other anti-angiogenic and neurotrophic molecules, which I don't have time to go into today. So I've shown you that we can take adult bone marrow or cord blood-derived endothelial progenitor cells. They'll target activated glia, and they'll participate in both normal and pathological angiogenesis. These cells themselves can stabilize and restore damaged vasculature and hypoxia and inherited disease models of retinal degeneration. And these cells mediate a profound rescue effect, largely by inducing upregulation of heat shock or neuroprotective molecules in the photoreceptors themselves, and also by inducing upregulation of a variety of other molecules. These are currently in clinical trials by a number of groups at UC Davis, Johnson & Johnson, England, Ireland, and Germany, and we await to hear what happens with the results of these clinical trials. Let me just finish up in the last 44 seconds. I may have to speak a little fast. Am I speaking too slow? So we've also turned to sort of disease in a dish. Kevin Eade has been heading this program. We've used retinal organizing. You'll hear more about this later this afternoon from one of the other speakers, not about our work, but about work being done with retinal organoids, in which you can actually take IPS and ES cells and differentiate them into form literally little eyeballs that grow a little in culture. And these cells can be analyzed for looking at functional. We can develop functional photoreceptors and muliglia, as evidenced by staining for muliglia or photoreceptor-specific antigens here. We can use, as you'd heard earlier, with targeted mutations. We are interested in a disease called macular telangiectasia, and it turns out there are a number of polymorphisms in genes involved in a variety of different metabolic pathways, and we can model this in these cells, which are grown. We now have about 30 cell lines from patients with MacTel and appropriate controlled patients, and they can be analyzed to do carbon tracing. Mary Gattner at LMRI has been looking at this. We can also look at functional metabolic analysis using seahorse type analysis, and basically explant. We can't take human eyes with disease in tissue culture, but we can make IPS cells from this. So I think as we go forward into the clinics with this process engineering, what's the best way to deliver RPE and these other cells to disease retinas? Do we do intravitreal injections? Do we do sheets? Do we inject? A very important question, which Valentina began to address, is can we make RPE from everyone's iPSC? That is, are we going to get the same kind of iPSCs from elderly patients? So patients with macular degeneration tend to be older. And if we want to do autologous grafts, can we get the same quality of cells from those patients we have from others? And of course, safety, safety, safety. We and others are looking at very significant proteomic, metabolomic, transcriptomic profiling these cells. Well, so what can really go wrong? This gentleman sitting here reading the newspaper saying, well, what is everybody worried about? There's nothing abnormal about this kind of thing. Well, we tried an experiment in the lab recently, and I actually volunteered to receive some of these cells just to make sure there weren't any adverse events. And unfortunately, the initial results was not quite what we expected. But as you can see, we had a way of reversing this. We had suicide genes and turned those on. So everything's fine. So I think there's been significant progress in understanding of stem cell biology and how it may be applied to problems in the central nervous system. I think our therapeutic options have expanded greatly, but they're not without limitations. You have to have the right diagnosis, you have to intervene at the appropriate time, and you have to think about what the microenvironment is in the eye as you put these, or other tissues as you put these cells in. I think they represent a great therapeutic opportunity, but there's also a great therapeutic challenge in terms of determining what cells you want to use and what are the conditions you want to use them under. And finally, we need to turn to our colleagues in biomedical engineering to better engineer cells and tissues which we can then put in. Our laboratories are now focused on targeted cell-based delivery. We've done this with encapsulated cell therapies and not only using the cells for the endogenous trophic activity, but also for the and to deliver neurotrophic and angiostatic molecules. We think extensive use of transcriptomics and metabolomics will help us better understand what is actually going on in response to cell-based therapies. And we think also as we identify more of the biomarkers, this will enable us to identify populations that are going to benefit better than other populations for this sort of therapy. And again, as you heard earlier, I think it's absolutely critical we better understand neurovascular glial trophic interactions, because that's really where the action is. You're not putting in one population of cells, it's only affecting one population of cells. Obviously, I don't do anything anymore in the lab. My kids say I just go to the lab and I talk on the phone and boss people around. So these are the people who actually did the work, and I've referred to them throughout the talk here. And you can't do anything without money, so we've been very fortunate to have great funding from National Eye Institute, Lowy Medical Research Institute, and CIRM, of course. And for those of you young people looking to do postdocs in pretty places, I don't have to tell you, that's our laboratory. That's Scripps over there. That's it. Okay, and I think I'll... and you'll never get onto the golf course to play golf, but you can watch them play golf from our laboratory with us. Okay. So I think I'll stop at this point, and thank you very much.